Hello friends, welcome to Teach Tigers. I'm Usha, your bio mentor. So ready with another session in human health and disease. So before going to our today's concept, let us see what we have discussed in our last class. So let's have a quick recap of what we have discussed in our last class. We have completed about AIDS. So we discussed what is AIDS. So what is AIDS here? It is acquired immunodeficiency syndrome. So we are acquiring, it's a sort of acquired immunodeficiency where the deficiency of immune system takes place, which leads to what? Syndrome. Syndrome is group of symptoms. So when our immune system is completely damaged, we are going to get so many sorts of symptoms because of various sorts of disease. So when our immune system is weak, definitely our body will be prone to get so many sorts of disease because we don't have the ability to fight. So that is what is happening with the AIDS. Then who is going to cause AIDS here? The virus. Which virus? It is retrovirus, by name human immunovirus. So it is HIV, human immunodeficiency viruses. Those are retroviruses which cause the disease AIDS. So we have discussed all about the AIDS concept like transmissions and symptoms and prevention and cure, right? So let's go with today's pick, that is cancer. So cancer, which evokes fever in mind. Yes? Why not? Because it actually it is taking millions of lives all over the world. Alone in India itself, it's taking most of the lives of the people. It is a silent killer. Cancer is a silent killer, which is a disease that kills many people, and because it is not even identified in the initial days, that's why it's called a silent killer. So cancer is a disease which is caused due to uncontrolled division of cells. So it is caused by uncontrolled division of cells. So scientists, what they explained is, this cancer is actually a disease which is caused by the loss of the control on the cell cycle and cell division. So it is basically a condition of failure to control the cell division. What is cell division here? So if you remember the cell chapter in +1, so cell and the cell cycle, we have for each and every cell we have a cycle, a programming, right? So where to divide and where to replicate, where to grow and increase in size, and where it is going to replicate and division. So all these we have a proper programming for each and every cell, that is called a cell cycle. Remember that? So that cell cycle and cell division will be interrupted or mutated, or there will be slight change in that because of various means. We'll discuss that also. So because of that, there will be failure of control to cell division. So that's why what happens when there is no control for division, they'll grow rapidly. Yes, so they will grow rapidly like this. When they rapidly grow, definitely there will be impact on the body. Yes, see, normally this is how our body looks like. This is the end of the growth. If the cell is required to, for division, where slight cut happens or somewhere where we need division, there it will divide. But do we have any uncontrolled division in our body? Generally we don't. But on exposure of few carcinogens, we can get some tumor cells. We'll see what they are. So unchecked division of cells leads to the formation of neoplasm. Unchecked division of cells leads to formation of neoplasm. Neoplasm is nothing but what? It is tumor. So neoplasm means tumor. So unchecked division of cell leads to the formation of neoplasm, that is tumors. So this sort of uncontrolled division or proliferation of cells leads to the formation of cells, that is called as neoplasia, which is nothing but formation of tumors. Okay. So proliferation means what? The word proliferate means to divide rapidly. So over division, that leads to neoplasia. Let us see what are the different types of tumors. Okay, tumors which are there. So tumors are actually of two types. One is benign tumor and the other one is malignant tumor. So the neoplasia or neoplasm may be of two types. One is benign tumor and the other one is malignant tumor. So what is this benign tumor? So can you see this picture clearly? This is benign tumor where the cell is, see, these are the normal cells and this is the cell where it showed some uncontrolled division where so many cells are present inside. This is the uncontrolled division. So here the tumor is formed, uncontrolled division is there, but does it causes cancer? No. It is a non-cancerous cell. Okay, it's a non-cancerous cells. Why it is non-cancerous cells? Because cancer is a movement. You know about the cancer, zodiac sign Cancer, right? What is there in that sign? Crab, right? So that crab, if you see the crab walking, we'll think that crab moves in this way, but crab moves in the other way. So that means what you think, it will not do that. So the same thing works with the cancer, with malignant tumor. I'll tell you what in detail also. So why it got the name cancer? Because of the movement, the walk in undifferentiated. So you will not think in which way it moves. It moves in irregular way. Okay. So here these are tumors, division takes place, but it is not cancerous because it will not move from one place to another place. Okay. It will stay there where the tumor is formed. Where the cell division is there, there itself it will stay. It will not move from one place to another place. So you can understand what is other tumor. Yes, what is the phenomenon of other tumor? What is the way of the other tumor? We'll see. So the next one is malignant tumor. You can see that here. This is also an uncontrolled division of cells only. But what happened here? The cells which are dividing inside, they are coming out. They are coming out from their source, and they are moving to various places. So these are cancerous cells. They cause cancer. These are the cancerous cells, and this is very, very deadly cancer because it spread from one cell to another cell, and it moves from one place to another place into tissues, into the bloodstream, through bloodstream to various parts, and even it affects various organs also. So it is difficult to predict where it is going and which cell it is going to affect. Understood? So that's why it is cancerous tumor. So let's see the picture here. So you can see this is how the cancer cells divides in between the normal cells. So these cancer cells will fight along with normal cells, and they will compete with normal cells for nourishment. Because when the cell is dividing rapidly, definitely when we are growing, when we are doing more work, we need a lot of energy. Yes. So here it is doing the division work. So it's also a work where it needs much energy. So from where the cancer cells will get energy here? So they have to compete with others. That other cells are nothing but normal cells, which are normal, which are not cancer cells. These cancer cells will compete with the normal cells and take away the nourishment. Okay, the nourishment will be taken by the cancers from the normal cells, and by that they grow rapidly. So now what happens is they will even move to various parts. You can see that they'll enter into various parts of the cells, and from there they'll enter into bloodstream, and they reach different parts of the body and even affect the organs. So the uncontrolled division of cells called as neoplastic tumor, which is nothing but a malignant tumor, and they invade the normal cells and cause starvation. So when the food is not available to normal cells, will the normal cells be alive? No. When there is no food, we cannot survive. The same thing goes with the normal cells also. When there is no food, no energy. No energy, it will starve. So it will starve to death, right? So cause starvation of normal cells. And not only that, this malignant tumor exhibits a frightening property of moving or invading from one cell to another cells and affects various tissues and organs of the body. That frightening phenomenon or property is called as metastasis. Metastasis, movement from one place to another place. This is about the malignant tumor. Let us know more about this cancer. So, the study of cancer, or the branch of science which deals with the tumors and including the study of how they grow, how we can diagnose, how to treat, and how to prevent. So all these study which is included about the cancer is oncology. So the practitioner who deals with oncology is oncologist. Okay, so this is about the study. A branch of science that deals with cancer is oncology, right? So next, let us see why actually this cancer is caused. Okay, how it is actually caused, how the cells are trying to grow. I already told you that entire cell is having some programming. So programming means when to divide, how much to divide, where to divide. All these have been programmed. Yes. But here, there is not a program here. That means they are not following the program. They are not following the sheet or charge what they have to do. So there is a slight change in that regular phenomenon. So we'll see what is that. So this is a normal cell. You can see clearly that normal cells, okay, they will have some integral adherents, okay, integral adhering junctions in between them. So these junctions are having E-cadherin, okay, which is present in between them, and they will make the cells to be intact with each other. So they will have a phenomenon called as contact inhibition. So they'll have a phenomenon called contact inhibition. So contact inhibition is nothing but inhibition of cell growth, cell motility and cell division. Okay, when they are in physical contact with each other, that means one cell is attached with or associated with another cell. So they will have phenomenon called as contact inhibition. So because of this contact, the growth of the cell and the division and the motility, movement will be under control. Okay, but what happens here in our cancer cells? So they will lose the E-cadherin, that is, there will be a mutation in E-cadherin. Because of that, they lose that property of contact inhibition. When there is no contact, and there is no one to tell that, okay, you have to stop division, or else now no need to move from one place to another place, then what happens? The cell will grow rapidly. If there is no one to stop, definitely it will do whatever it likes. That is what is happening here. Because of mutation in E-cadherin, here the cell adhesion is disturbed. Because of that, what happens? The disrupted cell-cell adhesions, you can see here, because of loss of E-cadherin, which is present in the normal cell, is missed here, and this leads to loss of contact inhibition. So this leads to lack of contact inhibition. Because of that, the cell will grow rapidly and vigorously it will proliferate and leads to the tumor cells. So this leads to what? Tumors. Got it here? Understood up till here? How the normal cell is becoming into tumor cell? Yes? So this is about contact inhibition. Now let's see more. So there are few genes in our body and there are few cells in our body which are actually, few cells are there which are like, which are called as proto-oncogenes. So proto-oncogenes are nothing but, there are few genes, you can see that. There are few genes which are just normal cellular genes, they will divide normally. But when these cells, okay, when these cells on exposure to any mutations or any radiations, you can see here, the cancer causing agents on exposure to any cancer causing agents such as radiations, either chemicals or any biological agents, then this normal cell will change into cancer cell. How it's going to change? You can see this is how the normal cell look like. On exposure to cancer causing agents, this proto-oncogene, oncogene is nothing but again cancer causing cell. It's a cancer causing cell. So in our body we have normal cancerous cells also which are in inactive form. When they are exposed to any cancer causing agents, they will get activated into cancerous cells. They'll be activated into oncogen cells, oncogenic cells. So because of that, what happens? Normal cell is going to become into cancer cell. Got it here? So normal cell is changing to cancer cells on exposure to cancer causing agents. Because of this, what happens? Normal cells which are in normal way, that is non-cancerous, are changing the nature into cancerous cells, and because of activated oncogenes, they are changed into cancer cells. So DNA got damaged and the proto-oncogene gene, again gene present in the genome, the cell nucleus, right? So that gene gets affected and activated. So because of this activation of this proto-oncogene, this gene will be activated as an oncogene which becomes into cancerous cell. So normal cell changed to cancer cell. Got it here? And then there are even few tumor suppressor genes also in our body. So there are few tumor suppressor genes. You can see that gene is gene P53, which is nothing but protein 53, which are responsible to make the cell to be normal state. So the name of this gene is nothing but tumor suppressor gene. What is tumor? It's a proliferation of cells or rapid division of cells, right? So cell division where it is there, that is called as, that will lead to formation of tumor, over bulgy structures of cell divisions, like that, growth. Okay, protruding cells, protruding skin, that is nothing but tumor. So they are having some genes which are responsible to suppress the growth of tumor. How they will do? As I already told you, cells will have the cell cycle. Yes. So you guys remember that G1, G2, S phase, like this. You remember all this, like G0 phase, M phase, mitotic, S phase, synthesis phase, gap 1, gap 2. Yes. So here what happens is, these P53 gene, protein 53 is a gene that codes for protein that inhibits the development and growth of tumors. How it will do? So this is a cell, okay, normal cell, which is having the DNA inside the nucleus. So this DNA, if it is damaged, then what happens is, if when a DNA is damaged for a normal cell, it's a sort of, see, generally this happens in our body also, some cells will get damaged, the DNA, gene or DNA gets damaged. Then what happens is, the genes like P53, it's going to find out this damaged gene and it will give time and it will help the normal cells to change or repair. It will give chance for the repair of what? The damaged DNA. If the repair is fine, if the repair is done, then what happens? This P53 will allow the cell to enter into S phase here. So after G1 phase, it will allow the cell to enter into S phase, where S phase, what happens in S phase? The replication takes place now. So replication takes place in S phase. If there is no division, if there is no repair is not done for this cell, so the DNA is not repaired, then what happens? The cell will be arrested in G1 phase itself. That means G1 arrest takes place and it will let this cell to undergo a programmed cell death called apoptosis, which is nothing but programmed cell death, apoptosis. So what this P53 is doing? If it will find out any cells are having any damaged DNA first. If there is damaged DNA, then it will allow and help the cell to repair itself. If it is repairing itself in a proper way, it will allow the cell to enter into S phase from G1 phase. Okay, if it is not repairing itself, if the DNA is not repaired, then what happens? This P53 gene will arrest the cell in G1 phase itself and it will not allow the cell to enter into S because the DNA is showing some changes. Because it's repaired in the sense what? Damaged in the sense it can show any mutations, it can show some chromosomal aberrations. So many can be there. So if it allows, what happens? When it enters into S phase, it will replicate in the same way. So damaged DNA is going to again repair into damaged DNA into so many cells. Yes or not? So that's why it has to stop there itself. And if it is not repaired, then the cell will be forced to undergo programmed cell death, which is called as apoptosis. Programmed cell death. Again, I'm using the word program because it's all programmed. If cell is unable to divide properly, then it will be undergoing apoptosis. That means it has to die on its own. Yes. So, if it is working, if the DNA is fine, if the damage is fine, then the cell will be allowed to enter into S phase where replication takes place. Okay. This is about the tumor suppressor genes. Understood here? So next, let's see what are the agents that cause cancer. So the agents that cause cancers are called carcinogens. So the cancer causing agents. Cancer causing agents. Okay. So what are those agents? Let's see what is the definition of it. Any substance that has the potential to cause cancer in living tissue. So in living tissue, any cells, okay, any agent, any substance that has the potential to cause cancer then it is called as carcinogen. So let us see what are the carcinogens, what types of carcinogens we have. So first one is physical carcinogen. So coming to the physical carcinogen. So in physical carcinogens, the examples are radiations here. So radiations such as UV rays, gamma rays. So this sort of rays, when they are exposed to the normal cells, then these radiations are exposed onto a normal cell. Then what happens is, this normal cell, this is a normal cell. Now radiations got affected on this normal cell. Because of that, again, the genome which is present, the DNA which is present inside the cell's nucleus gets disturbed. It gets damaged. And when the damage happened, you can see the DNA got damaged. Because of the damage, the cell will lose its ability to stop dividing when it has to stop. And because of that damage, because of the inability to stop division, this cell will become into cancer cell. So one cell became cancer cell, and what happens? That cell will rapidly divide. And I told you it will take the nourishment from the normal cells. It will compete for the growth. Yes. So because of that competition, what happens? The nutrition from the normal cells will be taken by this cancer cell. And then cancer cells will grow. The tumor cells will be formed. This leads to the formation of tumor. Understood, Ra? Yes. So let's see what are the other carcinogens that we have. So next, chemical carcinogens. Coming to chemical carcinogens. What type of chemicals can cause carcinogens? Few examples we'll take. So you can see here asbestos cancer. Okay, asbestos. So because of inhalation of the asbestos fibrils, what happens is they will block the lungs. They'll block the exchange of gases in the alveoli. Because of that, they can be inhaled, okay, through the nasal passes, and they will go and get deposited in the alveoli here. Because of that, what happens if something is obstructing the diffusion process, definitely there will be no exchange of gases. If something is obstructing the exchange of gases, then there will be no proper inlet and outlet of gases. That means there will be no exchange of gases. When there is no exchange of gases, definitely there will be suffocation taking place, and these asbestos fibers limit the exchange of oxygen into the bloodstream. So they will not allow the oxygen to enter into bloodstream, where there will be no proper movement of gases. So this leads to suffocation of lungs and even damage of the lungs. So this is about asbestos, inhalation of asbestos. And not only that, so you guys know about tobacco. Yes, cigarettes, that. So in that they have, in tobacco, there is a chemical called as nicotine. So this nicotine even have the chemicals, the chemical property or the chemical nature that can cause cancer in the lungs. So lung cancer can happen because of asbestos and nicotine and few of the chemicals which can cause cancer in the body. These are the chemicals which are acting like an agents and cause cancer. Got it? Yes. So next we'll see other type of carcinogens, that is biological carcinogens. So when it is physical, you thought it's related to some physical things and all. When it's chemical, you can understand that it's about the chemicals. But when I said about chemical and biological, when I said about biological, here bio means life forms. Yes. So life forms, that means there are few agents, biological agents that are causing cancer. So what are the cancerous causing biological agents? So, see what is this part actually? This is the cervix. This is the cervix of the female uterus. Yes, and this is the cervix. So, you can see that near the cervix part, okay, at the junction of this vagina and cervix part, you can see that there is some growth of cells. It's blocking the cervix. So, there's nothing but the tumor here. So, if you see from the below way, that is the cervix is completely infected with the cancer. So, because of the cancer cells at the junction of the cervix, it causes cervical cancer. How it is caused? It is caused by one of the biological agents, that is viruses. So, cervical cancer in women is caused by virus. What virus it is? HPV, human papilloma virus. So, this is one of the biological agents that can cause cancer in females. That is example cervical cancer. Okay. And how can you detect this one? By pap smear test. By pap test, you can say, or pap smear test, we can understand that the female is having the cervical cancer. Okay, by this detection, by this scan. Okay, so this is about biological carcinogens. Let's see more about cancer. So types of cancers. Coming to types. So we know what are types of tumors. What are types of cancers? Means which part is affected. What is the name of that cancer according to that affected part. Let's see one by one. So first one, if you take, it's an epithelial cancer here. So epithelium, epi, above, thelium, layer. Okay, so epithelial cancer is the cancer which is caused in the outermost covering of the body, right? All over inside, internally also we have different types of epithelium. We have discussed in structural organization, right? So there are different types of epithelial tissues where they are covering like a protection. Yes, and these are responsible to divide wherever damage happens. So that's why the division will be more in this epithelial cells. So it is most common to get the epithelial cancer. So carcinomas. Carcinomas are nothing but the epithelial cell cancer. Okay. So you can see here, these are the normal cells, how they are present in the epithelial layer. So if one of the cell get mutated, that exhibit oncogenic expression. Oncogenic expression is nothing but cancerous property. Yes, I already told you before on protooncogenes. So this oncogene expression is exhibited by this mutated cell and it will start dividing. So what it leads? It leads to division, proliferation. Because of that proliferation, so what happens? Our cells are tumor intelligent. You can ask me, madam, whenever such thing is happening, what will our immune system will do? Our immune system will try to recognize these cancer cells up till some extent, but because here they are taking the nourishment from other cells, because of that the energy will be reduced by the normal cells and it will activate the cancer cells to divide more and more. So you can see here, so it leads to carcinoma, okay, the tumor formation epithelial tissue. So because of that, we can see some small projections, nodes like structures on our body when we get a epithelial cancer. Okay, and next one is lymphoma. So lymphoma, lymph, we have discussed somewhere about this lymph. Lymph is actually a fluid, whereas we have discussed about lymph vessels, lymph nodes in immune system. Yes, primary lymphoid organs, secondary lymphoid organs. I told you that lymph nodes, lymph vessels comes under secondary lymphoid organs. Yes, so they are related to immune system, na? Resistance, production of lymphocytes, B lymphocytes, T lymphocytes. Did we discuss? Yes. So if the cancer is caused in the lymphatic system, when the cancer is there in the lymphatic system or affects the lymphocyte, then it is called as lymphoma. So cancer of lymphocytes. What are the lymphocytes we have? We have B lymphocytes, which is coming from the bone marrow. Yes. And T lymphocytes, again, synthesized in the bone marrow but get matured in the thymus. You remember that? So these lymphocytes can be affected because of this lymphoma. Okay. So what happens is cancer cells must develop in thymus. Again, thymus is a primary lymphoid organ. Lymph nodes, secondary lymphoid organs. Bone marrow. Bone marrow is again primary or spleen is again secondary. So these are the primary and secondary lymphoid organs where the cancer cell may develop in that particular lymph nodes. Because of that, the tumor will be formed. So when the tumor is formed, tumor means what again? Division of cells. So when the division of cells takes place, what happens here? It leads to cancer. Yes. So this cause cancer in lymph nodes and lymphocytes, and there the cells will divide and they become into tumors. This is one more type. And we'll see the next one. Leukemia. So leukemia, leukocytes. We already know about leukocytes. They are nothing but WBC cells. You can connect like that, right? So the cancer that cause in the blood, that is in blood cells. If the cancer is caused, then that is leukemia. So what happens here? The increase whenever the cancer takes place in the bloodstream, develop in the bloodstream, then I already told you that the policeman of our body, that is WBC and neutrophil cells, there are so many cells in the leukocytes, right? So those cells we'll identify at wherever the spot the abnormality is there the number of cells increases. And here in the bloodstream, because of the cancer cells in the bone marrow, okay, it happens in the bone marrow. Bone marrow is a place where synthesis of all the cells takes place. It is a hematopoietic stem cell. It's a stem cell, that means division of cells and production of cells, RBC, WBC, platelets, all takes place. So there in bone marrow, if the cancer takes place, then what happens? It will result in unrestrained production of WBC. So more percentage of WBC cells takes place. So it's a sort of liquid tumor. Okay, it's a sort of liquid tumor because in blood it is taking place. The number of cells are increasing. So this is about leukemia. Understood the types of cancers? Yes. Now let's see what are the ways to detect. So how to detect the cancer? So detection of cancer. So how we can detect? Now you know where the cancer takes place, what are the types also. So just now we have discussed about leukemia. So leukemia is a cancer where it takes place, cancer cells will be developed in the bone marrow here. So where the cell division is there in the bone marrow. Now how to recognize if the cells will increase in bone marrow. So what we can do? The detection is biopsy. So biopsy. So in biopsy what happens is we'll take a small piece of the tissue. Okay. Then we will check about the tissues, the growth of cells and all. So what we'll do is, to check the biopsy, to do biopsy, we'll take bone marrow aspiration needle, like this it will be there, where this needle is being injected into the bone marrow and then the cells are being collected. The marrow is collected, then they are going to check the smear of it, okay, and they will see how the cells are dividing. So that is detected by histopathological studies. So they will take the smear, they'll take the tissue and they'll do the histopathological study. What is histo? Histo means tissues. Checking out the pathogens. So histopathological study is being done and they'll detect the cancerous cells which are present in the bone marrow or not. So even they can go for blood test also. In blood test, definitely cancerous cells will be flowing in the blood. So in the blood cancer, in leukemia, we can go with the blood test. If it is cancer in high level, then we can go with biopsy, bone marrow aspiration or biopsy. And there are few more detections also. The next one is computed tomography. So CT scan. So where the in the CT scan, the X-rays are used. X-rays are used to detect the cancerous cells that are produced in our body. So this detection of cancerous cells or detection of any disease, okay, that is computed tomography scan. It's where the X-rays are used, okay. The next one is mammogram. So mammogram, mammary glands, that is if the female is suspected to have breast cancer or there is some tumor type of appearance in the breast region, so she can go for mammogram. So this is a detection test where medium or low intensity X-ray beam is being exposed on the breast region, on the mammary glands, and then the detection of cancer is being done. Okay, this is about the cancer detection. We have few more like MRI scan. So in MRI scanning, you can see that the entire body is being exposed to magnetic, okay, resonance imaging, where strong magnetic fields are used and even non-ionizing radiations are also used to detect the entire, okay, the cancerous cells or any disease in the body. So this is how we can detect the cancer. So now we have identified here. So what is a way of treatment? So how you can treat if you have detected the cancer? So the treatment of cancer is one by surgical treatment, surgical way. So you will do surgery and remove of malignant tumors. So in surgery what they will do is, they will identify the tumor cells. The tumor cells are being surgically removed. So they will cut this part and they will remove it. So this is one way. And the other way is radiation treatment, where the radiations are, okay, used and the radiations are exposed on the individual who is suspected with the cancer, and then by using radiations we can treat the cancer. This is one more method, and we have few more treatment methods, that is biotherapy. So in biotherapy, what they'll do? Biotherapy is nothing but it's a sort of prophylactic cure here, where once the cancer cells are increased in our body, our immune system will become weak. Yes, when it is weak, definitely what happens? Our immune system cannot detect the antigens. Here antigens are nothing but cancer cells itself. So when our cancer cells are not detected by our own immune system, that means our body is in weak condition. So to activate the immune system, what they'll use is biological response modifiers. They will activate our immune system by using alpha interferons. These alpha interferons are biological response modifiers. So they are being injected into the body, and then they can have the ability to identify the cancer cells and destroy that. Okay, this can be even done after the surgery to find out if any leftover, because I already told you, in malignant tumors, the cell can invade into other cells. So though you do surgery also, there will be a chance of the remaining cells, the remnants of few of the tumor cells. So at that time, this alpha interferons will work. This is a sort of biotherapy. Okay. The next one is a chemotherapy. In chemotherapy, what they'll do? So they will introduce chemicals. You can understand, chemo means chemicals and drugs. Okay, these are of high intensity or high level drugs where they are injected into the body, and because of these chemicals will identify the cancer cells and they will destroy the cancer cells. But what happens, because of these chemicals will have high side effects such as a loss of hair fall. Okay, hair will fall off, and eyebrows will fall off. So there will be no hair on the body. This is one of the side effects, and even anemia. Blood loss also takes place. So these are some of the side effects for chemotherapy, and chemotherapy is also done even after the surgery. These are all the sequential treatments here. These are all the step-by-step process of treatment in cancer, because as I told you, in malignant tumors, these tumors, we cannot say that once we have done the surgery, the entire cancerous cells are removed. We cannot say, because it exhibits a phenomenon. I already told you, it's a scary property, that movement of these tumors from one cell to another cell, it takes place. They will invade through bloodstream and enter into various organs also. It can enter to various places and affect those cells also. So because of that, what happens? We have the chance to get cancer in any other place. Any organ can be damaged. Yes. So to identify that cancer, it is difficult. So all these treatments in a stepwise process will have major impact on the cancer treatment, and then we can reduce the cancer. This is a sort of cure that we do. Okay. So this is all about the cancer, guys. So I hope everything is clear. End of a concept. So what we have completed up till now, please have a quick revision and get ready with another new concept in human health and disease in our next class. Until then, bye-bye.